I'm not going to talk about specific research findings today, except by way of showing an example of what you can do if you share data across very large data sets. But I would like to emphasize with the people here who want loud voices of advocacy that Canada has an extraordinarily rich history of contributions to Parkinson's research. And we have consistently, as a country, we have consistently punched way above our weight. That includes participation in the discovery of dopamine deficiency, the early use of levodopa, understanding of basal ganglia circuitry, actually in imaging and in genetics of Parkinson's. So people in Canada should feel very proud, but times have changed, and so there's lots more that we can do now by rethinking the way we do research. So you'll recognize immediately which are the slides that I created and which are the ones that were handed to me by Parkinson's Canada. This is one of the latter. This is the history of the network. And so it really came out of, as Joyce already alluded to, an idea that was born in Quebec. So Quebec formed their own Parkinson network. And Quebec, I would say, has an extremely distinguished history in Parkinson's, which then went a little bit fallow for a while. And they have reactivated themselves. And it's not only because of the network, but with the network, they have been incredibly productive. And so are now major players in the field. So in early 2016, Uri Monchi was, I can't remember whether he'd moved by then or not. I think Uri moved from Montreal to Calgary, which was, I guess, part of the same province. And he thought that it would be a good idea, talking with Ted Fawn in Montreal and me, if we could sort of join forces across the country. And so we had this meeting. I think this was in the lead up to the US election, because I remember watching debate number 1 in the lobby as we were walking past. And so there are a number of people here who were all very keen to see this move forward. And we did require a fair bit of work to put together a grant application. Brain Canada is an organization that funds brain research, and they had platform grants. But Brain Canada was set up so that match funding is required. And Parkinson Canada not only provided the match funding, but actually has really led the cause here and has been very actively engaged in facilitating initially the formation and now the actual development of the network. And just as an aside, I would say that Parkinson Canada, like other organizations of that kind, goes through good years and bad years. And Joyce, in her role as CEO, ensured that the research budget was always protected no matter what else happened. And for that, I and the rest of the Parkinson community are very grateful. So what is the network about? Well, the idea is to bring together the best people in Canada who are working on Parkinson's, but rather than having them work in isolation, share data so that we can each take advantage of each other's work. The history of research, of course, is one where many of you may envisage some brilliant person who's incapable of interacting with society, sitting in a little garret somewhere in an ivory tower, having brilliant ideas, and those ideas immediately come to fruition. Well, it's probably actually never worked that way. There will be the occasional genius who functions very well in that setting. But we have always depended on people sharing expertise, and many of us work in centers with other disciplines where we can learn from people who come at the same problem from a different perspective. But we now have totally different information available to us, and what I'll show you later is that we're at a stage where no one center, let alone any one individual, can possibly extract the kind of information that is available to us in isolation. So we really depend upon our ability to share. So this will, of course, facilitate collaborations, and I'm not going to talk much about clinical trials today, but as one advantage, when new treatments are introduced, they have to be tested because it actually turns out that people aren't quite the same as mice. And so it's much more difficult than you might think to recruit people to participate in clinical trials, because most of these trials have fairly strict criteria for who gets in, who doesn't get in, and that's often for the protection of the individual who's participating, because if you have bad heart disease or you have a history of cancer, it may not be such a good thing to give you a new treatment that's being looked at for Parkinson's. So it's hard to recruit. Often the trials are quite demanding with frequent visits. If you have a registry of people with not only defined according to diagnosis, but also according to complications or the basis for the diagnosis, it's much easier to recruit. And so if you're a researcher in Edmonton and you say, well, I'd like to do such and such a type of study in people with Parkinson's, but people who have depression and an impulse control disorder, you'd be hard-pressed to attract enough people if you just go into flip through charts. But if you can actually go into a registry without identifying people by name, you can say, oh, so-and-so in London has 100 people like that. And we know that there's another 50 like that in Quebec City. Then you can pick up the phone, contact your colleague and say, this is what I want to do, can we work together on this? That is an enormous asset. That will be an enormous asset not only for the scientific research community, but also for pharma and biotech companies who want to be able to access patients. This is extremely valuable for them. And of course, this allows much larger scale projects that could not possibly happen at a single site. And I'll show you some examples of that. So as I already indicated, this is really modeled after a concept that was born in Quebec. And in a very short time, I don't recall when QPN was founded, but they ran more than 50 studies making use of a registry. So people come into the clinics, it's a simple registration process, and then a more detailed questionnaire that follows. And they've registered more than 1,000 patients. And from a research productivity perspective, they've generated a lot of publications just with this fairly recent network that they formed within the province. It has challenges, there's no doubt about it, but they've been very successful at it. So I think this slide says what I just said, but there are some other points here, and I think we heard that point made in the introductory comments this morning. Research is hope. Without research, we don't have new treatments. We don't understand the disease as well as we should, but additionally, there's all kinds of evidence to show that people who participate, who are actively engaged in their disease, which may include participation in research studies, actually do better simply by virtue of that level of engagement. And so coming in, knowing that your doctor is participating in something bigger than the clinic visit is a motivator for many people to go to that particular clinic, and helps you understand that people are outside the consultation, people are working and thinking about how to make the world with Parkinson's a better world. So the QPN has successfully connected patients, clinicians, and researchers, and have innovated something that we hope to see as part of the Canadian Open Parkinson Network, which is a web-based enrollment process so that patients can enroll themselves in a protected website, and will eventually even be able to complete questionnaires and so on. So the network is distributed more or less across the country. The red flags are for the existing sites in the initial network, and you can see this stretches from Quebec to British Columbia, but we still haven't covered the entire country, and there is certainly a hope to fill in some of the missing centers, and this has been limited by funding. The funding that's available at this point with Parkinson's Canada and Brain Canada combined is $2 million, which sounds like a lot of money, but this basically pays for most of a coordinator at each site. And that's basically it. It does not really pay for data collection, and some of the data acquisitions are extremely expensive. I won't talk about that. So why should we, now we're back to the slides that I prepared. Those are the simple black and white ones. And why share data? Because it's very difficult. If you're working, you think of that old cartoon of people feeling an elephant with blindfolded and trying to figure out what it is. Well, the person who's got his hand in one spot doesn't feel the same as the person who's got his hand somewhere else. And we tend to look at the factors that lead to Parkinson's from a perspective that is based on our own scientific training. And as science becomes more specialized, that problem actually, ironically, becomes worse, because people are ultra-specialized, they're superb at doing what they do, but they may not always have a broader perspective. And so when you bring people together and share the data, you're more likely to be able to have a broader perspective. A really important aspect, of course, people want a cure. I'm not going to talk about a cure except to say that it's not straightforward, or somebody would have done it a long time ago. So we still need to keep in mind what it's like to live with Parkinson's. There are a lot of complications of Parkinson's. There are a lot of complications of the treatment. And no two people with Parkinson's are the same. And so there are some people in whom the disease progresses fairly rapidly, other people who do well for many years. And we don't really have a good understanding of why one person has a relatively benign course and other people have a very difficult course. That's going to be very difficult to understand unless you can look at very large numbers of data. And so having a network permits one to address that kind of question. I'm quite sure that no single clinic could possibly hope to do that. And I think we've also tended to focus, people who work in health research tend to focus on why people get worse, which is an understandable focus, but there has probably been, until quite recently, inadequate attention paid to the factors that predict better outcomes. And if we understood why some people do better, that may actually be a more appropriate focus, or certainly one that should not be forgotten, because that may be very informative in general. I've already talked about facilitating participation in studies, in clinically oriented research, and in clinical trials. And another important factor that is often underappreciated is it's not straightforward to do these things. I do imaging type research. We have a variety of tools that we can use, and you would never tell it from reading the papers, because if you read the publications, it says, well, this is why we did it, we did this is what we found, aren't we brilliant? And not clearly elucidated in those papers usually are all the methodological challenges. And the truth is that many of us share those methodological challenges, or there may be more than one approach to addressing a certain question. And it's only if we can share data that we can better understand how to choose a more common approach, or how to overcome some of those difficulties. So I'm going to just show you some examples of how one can look at certain problems by sharing data. This is actually, the only reason I've put this slide up is not because of the data, which are not remotely interesting, but rather to highlight who the authors are. And you will notice that the first author is from Intel Corporation. Now, Intel historically has an interest in Parkinson's that I won't get into, but it's a shared authorship between Intel and the Michael J. Fox Foundation. And what they're looking at is the use of wearable sensors to try and address problems on a bigger scope. So, as you all know, if you've got Parkinson's, you come in for your appointment with the neurologist, you get a couple of minutes of quality time while they examine you, and they're getting a snapshot of what your overall function is like for the rest of the year. And that snapshot is going to be heavily influenced by A, where you are in your medication cycle, B, what kind of a day you're having, and C, the fact that it's stressful to be there. So it's not really a realistic view of what your Parkinson's is like. Much more realistic view would be if your neurologist would move in with you and examine you every couple of minutes. That's probably also not feasible. So having some way of measuring even motor function, which is a very incomplete measure, but if you could even do that, that would give you a much better idea of how things are going. And there are a lot of wearable sensors that are being developed. In fact, this can all be done. I've got a Fitbit on, and I've got an iPhone in my pocket, and those are all things, of course, that can be used to try and collect that kind of information. And some people have taken it much farther. I've got a colleague in England who actually works in a house that was designed by engineers, and there are videos. So you can actually watch people preparing their meals and as they go about their daily activities. And then there are tricks to anonymize this so that you're not really kind of creepily invading on people's privacy. There are ways that you can actually, in fact, my colleague in Vancouver, who's also an ex-Londoner, it has a device that will use facial recognition so that only the person of interest is being monitored, but the facial recognition is used only for activating the recording and is then not used. So that information cannot be used to invade privacy. And then what's recorded are actually stick figures as people move about the house. So this is just an example of using the wearables, and you can appreciate how much more information you can derive from this as opposed to the standard office visit, which still needs to take place, by the way. This is another one that came from a large cohort, and I think Fox Foundation may be involved in this one as well. So yes, it is, because this is from the Parkinson's Progression Markers Initiative. So they used data for the PPMI is a study that has registered 800 people with Parkinson's and 400 healthy controls in the US. They're subjected to very intensive investigations. And they've added some cohorts since with people who have risks, who are at risk of developing Parkinson's. And this is all done by a bunch of geeks. There are some neurologists in the authorship here, but mostly these are geeks who analyze data, and I can't even understand the abstract, let alone what they actually did. But I just want to highlight that they got some information that allows them to predict, just by using machine learning, so artificial intelligence, if you will, who has Parkinson's. Now, important information in there, thank God, is actually the clinical information, but there are other factors, so that if they combine them, and if you do this by traditional hypothesis-based science, you would have some difficulty coming up with this combination. So this is basically an example of how they just take all the information, feed it into a black box, and then the black box says, these are the things that together are predictive. There's been a lot of interest, of course, in the genetics of Parkinson's. As most of you know, most Parkinson's is not a genetic disorder. It's estimated that probably between 5 and 10% has a genetic basis in the traditional view. But there may be genetic risk factors that contribute to Parkinson's, quite likely in combination with various environmental factors. There's going to be lots of those as well. So there are two types of genetic study. One is where you identify an actual gene mutation. So you know if you make this particular change in a gene, that is going to likely lead to disease. That forms a minority of Parkinson's. However, if you look at very large numbers of individuals, variation in certain genes, which is not enough on its own to cause disease, may be associated statistically with an increased risk. And sometimes it's the same genes. So sometimes there's a mutation, in other words, a variation that is known to cause disease. And sometimes it's a variation in the same gene that doesn't cause disease, but statistically is associated with a higher risk. This can only be done, this approach to look at the increased risk is referred to as Genomewide association study. So they basically look at a large number of people who have the disease, a large number of people who do not have the disease, and then look at the entire genetic code and determine which variants are associated with an increased risk. This can only be done with very large data sets, and this is simply one of the more recent examples. I think this is a paper from 2017 where they replicated earlier findings from GWAS studies, but they also, because they used such large numbers, they were able to identify 17 new areas in the genetic code that are associated with increased risk. This is important because it points to certain mechanisms that are more likely to play a role, and in this case, I think they found a role for a process called autophagy, which is a process by which proteins are broken down, and lysosomes are little components in the cell that are responsible for breakdown of proteins, among other things. So it points to that direction. So that's really important in terms of understanding mechanisms of disease and developing new treatments. This just shows you another example of the power of large data sets. This is an imaging and genetic study that operates worldwide, and they're actually primarily looking at psychiatric disorders. But this was one of the first papers that came from the group looking at the influence of genetic variant on normal brain structure in healthy individuals. So we think if we're healthy, we understand that. But it turns out that there is variation in the size of different structures. And these two structures that are shown here are part of the basal ganglia. So they're affected in Parkinson's. The pallidum is where surgery is done for Parkinson's. And the size of these structures varies amongst normal individuals, and that may be predicted by certain genetic variants. The immediate practical importance of that is that if you're going to try and interpret the findings of doing certain studies, you may need to take into consideration the genetic variant of the individual, because that may actually have an influence. But there are certain genes that are associated with these variations, and in this case, it's genes that are associated with development of the nervous system and with programmed cell death. And so that may be giving us some insight into what actually happens in the basal ganglia, but you can only do this if you look at thousands and thousands of people. You can't do it in small studies that we typically do at single centers. The advantage of this, and I'm going to show it to you again in another slide, is that you can actually look at people with Parkinson's. So now instead of trying to do something to a mouse or a rat, you can take cells from people with Parkinson's and test a variety of interventions in those cells. And so these are cells from healthy control on the left and somebody with a LRRK2 mutation, a genetic mutation that causes Parkinson's on the right. And you can see that the cells from the patient with the mutation express too much of a protein called alpha-synuclein, which is the protein associated with Parkinson's. And then if they treat the cells with taurursine in a dish, they can actually revert those levels to normal. So that's a very elegant way of looking at this. And then they used the large data set. So I referred earlier to PPMI. They looked at people who were participating in PPMI, and they looked at those people taking taurursine, which nobody thought about, frankly, at the time. Nobody had any reason to believe that taurursine might have any impact. But because they had a reasonably large number of data, they were able to go back and compare the progression rates in people treated with taurursine, which is the red line, or the others, which are the green lines and blue line there. And you can see that there's a difference in the rate at which people get worse. Now, despite the fact that PPMI had a reasonable sized cohort, there were very few people taking taurursine. Taurursine has a mechanism of action. It's an alpha-1 blocker. So in order to deal with that potential confound, they looked at a different alpha-1 blocker, which is a drug called tamsulosin, which is also used for treating prostate disease. And they did not see that protective effect with tamsulosin. So the point here is, if it's taurursine or similar drugs, you do see this small protective effect. If you look at other drugs with the same mechanism, you do not, because there's a second difference in mechanism. So, but it's not very big numbers. And so now, just to show you the power of big numbers, they looked at a large insurance cohort or a marketing cohort, something like that. And now you can see they're looking at 15,500 people who were on taurursine against people who were not. And what they found was, if they looked at people with Parkinson's, the number of complications was significantly lower in the people who were exposed to taurursine or a similar drug, not in the people who were exposed to tamsulosin or in the control group who still had Parkinson's. And if they compared controls who were treated with taurursine against people who were not, they found that there was a lower emergence of Parkinson's disease in the people treated with taurursine. So should everybody be treated with taurursine? No, because it's a drug that can lower blood pressure. This needs to be replicated, and it's a drug that has potential side effects. But you could only get this kind of information by looking at huge numbers of data. For that, you need the cooperation of multiple centers. I already referred to the induced pluripotent stem cells. This is one of the plans of the C-OPN. The group in Montreal in particular are very interested in this. So the idea is you can get regular cells instead of going out and getting a biopsy of brain. You can get regular cells, convert them to these induced pluripotent stem cells, so you can do this from skin or from blood. And in fact, that's the plan, is every center in the country who's participating in this will draw blood and send the peripheral blood cells to Montreal, where they will then derive the stem cells. And one of the other points I want to make about data sharing is that it's not just sharing data sets, but it's actually sharing, if you want to take it to a higher level, you will share scientific discoveries as they emerge. So the Montreal Neurological Institute has taken a very aggressive position in this, and Guy Rouleau, who's the director of the MNI, is involved in C-OPN. He's basically suggested strongly that all new discoveries at the Neuro become publicly accessible as soon as possible. The second last author on this paper is Ted Fawn, who is an old friend. Working with him is Tom Durkin, who has taken this to a new extreme. Tom actually puts his lab notebooks available online at the end of the day, so you can actually follow what he's doing in real time. This is not from his lab notebook, but actually the day that Guy and I spoke about having an agreement between UBC and the Neuro on open science, I was talking to Guy on the phone and I thought I'd better check what this is all about, so I looked up open science at the Neuro, and guess what paper popped up that had just been put online that day was how to take these cells and then you make them not only into the type of cell you want, but you allow them to grow together and they form like a midbrain, the part of the brain that's affected in Parkinson's disease. So this may allow one to do yet further research that you couldn't do in individual cells. So I'm sorry for the time I've taken. I'm just going to finish up here by quickly reviewing the structure here. The Open Parkinson Network is led by Uri Monchi, who is now in Calgary. There is an executive committee, which I think is Ted Fawn, Antonio Strafella from Toronto, and me. There is an international advisory board. These are super scientists working in the Parkinson's domain, covering genetics, imaging, and some other factors. And then there are some scientific committees with various makeup. This all depends on the work of the coordinators at the different sites. And then we're still working on having an ethics advisor and patient advocates, which are an important aspect of this activity. So the idea is people will sign up either during a clinic visit or online, are connected to the closest site if they're doing it online. This requires some visits. The initial step is to just get registered, and there's only very basic information that's required. But then, of course, for people who choose to participate, there's a much more detailed questionnaire and some simple testing. And then it's a registry. So the information is put online, but in an anonymized fashion. That's obviously incredibly important because confidentiality and respect for privacy is a big issue. And then people can query the database. There will also be a biobank of biological samples, and I already indicated, and you actually heard from the introductory comments this morning, that people do believe that participating in research gives them hope. And so I feel that we should be doing research not only for our own interest and for the information that can be derived, but I think there's actually an ethical imperative to do research. And this is a testimonial from somebody who is unknown to me. And to just quickly review the goals. So I think we've talked about all of this. I would just say that it increases our dominance in the world field again in Parkinson's, increases our opportunity to participate in clinical trials and develop new treatments, and to really move discoveries from the bench to the bedside faster. So if you want to get involved, right now London is not a site, but there's obviously a hope that London will be. But in the meantime, it should be possible to get people engaged and linked to the closest site, which I guess for here is Toronto. As Joyce also indicated, this does require a lot more funding in order to engage more sites. So be an advocate, get out there and help Parkinson's Canada with the fundraising. And Julia's in the room, Catherine is not. And I think this was a recent meeting held in conjunction with the Movement Disorders Society meeting in France. And you can see all these really happy people who are just itching to get going. Thanks very much.